majority. All right, I will share my screen, and you guys are going to have to tell me when you want slides to be changed. Can you guys see the slides? Yeah. Okay. Give me a minute to make this. I don't usually present out of PDFs, so. Does this look normal too?
Yeah.
Okay. All right. Go for it.
Okay. So our group was assigned over Chapter 4, which was the agronomic and environmental effects of genetically engineered crops. Next slide, please. So to start off, as with all the other chapters, our chapter included a lot of experiments, and this was one of the first experiments discussed in the chapter. And in this experiment, it showed basically the distinction between the potential yield and the actual yield of corn. And the potential yield is the theoretical yield a crop genotype can achieve without any limitations of water or nutrients and without losses to pests and disease. So if you look at the graph, you can also see the potential yield, the water and nutrient limited yield, and the actual yield. To talk about the limitations of the natural nutrients and water availability, that's what causes the gaps between the potential yield and the actual yield. So if nutrient and water supplementation are not possible, then that's what's going to happen. So yeah. Basically, if you look at the graph, it shows the potential yield of production and the production of the water and nutrient limited crops, as well as the actual production of the crops. So, and what makes up these amounts is dependent on the factors next to the arrows that you can see next to them. So basically the main takeaways from this experiment, in the reading, it said that the genetic improvement of crops can close the gap between the actual yield and potential yield, or it could even increase the overall potential yield. They said it could be accomplished in 3 ways, and those ways were the potential yield can be increased by, it gave an example, it was the canopy architecture of the plant could be improved to increase the conversion of photosynthesis through active radiation. It was a little bit confusing to me, but it was definitely a possibility. Secondly, the limitations of water and nutrient can be reduced by enhancing the efficiency with water and nutrients are captured and used for crop growth. So basically with the GMOs, it can reduce the water and nutrient effects on the potential yield. And finally, the factors that reduce the yield can be mitigated by protecting crops from pests, including weeds, insects, and disease. So yeah. Next slide. So the next experiment that it talked about in the chapter was basically it relates to the first one. It was the effects of genetically engineered traits versus just regular plant breeding and on the overall yield of the actual plants, but this time it was over soybeans, corn, or soybeans, cotton, and corn. And so if you look at the graph, you can see the changes in yield of those crops from the United States in 1980 to 2011. And a good thing to note is that the dashed line in the middle of these graphs is whenever the genetically engineered crops were introduced. So basically, if you do look at that graph, then you can see that it wasn't really an overall effect on genetically engineered crops to the yield, but they've still increased over time. So yeah, that's next slide.
Okay, so the next few graphs I want to show you, they go with BT corn and BT cotton, sorry. And BT is where they're genetically engineered, so they were able to fight off like insects and everything without the use of insecticide or less use of insecticide throughout their life in the field. So this graph will show the rates of insecticide applied to the crops of cotton and corn in the US. There were different research groups starting in 1996 that studied whether or not the genetically engineered crops needed less insecticide or not. In one study by Klumper and Quam, I think it was, it showed a 39% decrease in the usage of insecticide. In another study in 2010 by the National Research Council just stated that there was a steady decline in the amount of pounds per crop when they were BT corn or BT cotton. Next slide, please. Okay. Graph 1 is identifying the usage of insecticide by the BT adopters and non-adopters. So the BT adopters, they'd obviously be using the seeds that were genetically engineered for the corn or the cotton. And then the non-adopters are just they're not using the BT seeds. Some people wanted to use the BT seeds and the others didn't, so that's how they kind of got their research going. And so that is showing that non-adopters were using more insecticide for their corn crops versus the adopters. So that first graph is just for corn, it doesn't show cotton. And then graph 2 shows the amount of insecticide used on cotton fields in China per season. The blue dots are total insecticide. The green dots are insecticides sprayed aimed at the cotton bollworm, so the actual insects on the cotton plants. And then the red arrow indicates the year when the BT cotton was first commercialized. So kind of when it went out to the public. Next slide.
So this graph shows the herbicide usage per kilograms and hectare between 1995 and 2010. So as you can see, they gradually decreased until about 2001, 2002, but the decrease isn't sustained. So they determined that it wasn't useful to determine the risk for humans and the environment. A couple other things I found was that as herbicide resistant crops were used continuously, applying the same stuff didn't get rid of 100% of the weeds. So there were still some coming through. And with that, the returns of the crops went down. And because the management tactics were pricey, and basically how they figured those things out were they had a couple fields in Michigan between 2002 and 2006, and they just figured the averages through that 5 years, and they determined that 2 had a better average with glyphosate than the other herbicides, and they had 2 fields in Illinois, and they found that it worked better in sensitive temperatures with moisture. And basically it just had to acclimate the first year to the second year. And a few takeaways from this was there's not really any research on cotton, but canola, sugar beet, and alfalfa showed that the HR had a bigger yield and weed control is improved than when it's not. That's it. Next slide.
So since 1987, there's been a decrease of in diversity crops in the U.S. And when we look at those crops, that's maize, wheat, and soybean, because the rotation has been so high, every single one of these rotated plants have been presented at 80% and up. So planting of BT variables of crops tend to result in higher insect biodiversity. And just lastly, no evidence was found of cause and effect between relationship, GE crops, and environmental problems from the group that was doing the research. And that is it.
So just to wrap it up, we put basically the main takeaways of this chapter on this slide. I know it's a lot of words, but it'll be helpful later. So yeah, just basically genetic improvement of crops can close the gap between actual yield and potential yield. That's just one of them, but another important one is that HR crops contribute better yield when weed control is greater than not. And then basically since 1987, there's been a decrease in diversity of crops grown in the US. So yeah, that's basically just the main takeaways over here. Yeah.
All right, questions from the crowd?
It's not necessarily related to the content of the presentation, but it might be helpful. Could we all get a copy of each presentation?
Yeah, I'll just need to get copies from, I have obviously this one, but I'll just need to get copies of the other presentations. I'll just post them all on Moodle, and then everybody can have them. Questions about the presentation from the presenters, or for the presenters rather. Really, you guys have no questions about that presentation whatsoever? When I surveyed you guys, some of you said that you wanted more discussion and things like this in class because you thought that you would learn better and it would make it more interesting. Now's your chance. Go for it. You didn't think I was going to read those survey results, did you? Okay, I'll get you guys started off. So Blake, you were talking about this figure, weren't you?
Yes.
So can you remind us what the significance of this figure is? Why would you want to pay attention to this figure? What's important about this figure?
Well, something I kind of realized I forgot to mention was that the different types of crops are actually given certain traits, and so corn and cotton were given the BT trait, which was that Bacillus stringus, I think, that we've already learned about. They were also given the HR trait, which is the herbicide resistant trait, and then soybeans were only given the herbicide resistant trait. So those were just.
Okay, but what's the significance of the line going through the data? Why did they put a line through the data like that?
Just to show like the increase over time and how there's really no substantial effect that they have on the crops so far.
Yeah, so and you were getting at that, but you didn't say it quite as clearly when you presented. So the thing that you would expect is after after the introduction of genetically engineered crops, you should expect the slope of this line to tick upward if it was actually increasing yields over and above what you would have expected just from changes in technology not related to genetically engineered crops. But you see that in all of these cases, the slope of that line doesn't change after the introduction of genetically engineered crops. So genetically engineered crops aren't seeming to affect yields at all. They increase after the introduction of genetically engineered crops, but they're increasing at the same rate as they were before you had genetically engineered crops. So it's not a crop yield issue why you might use these things, at least not according to these data. So what might be important if you're not getting greater yield, what would be an advantage of using these?
It lowers how much the crops are infected by the pests and things like that.
Yeah, so maybe you don't make more money as a farmer because you're getting higher yields. Maybe you make more money because you're having to use pesticides less, and pesticides cost money. And it costs money to apply those pesticides to your fields, and so that's less money that you have to invest in your crop because it has this genetically engineered trait. So you're selling the same, you're growing the same amount of crop, but you're growing it at a cheaper cost because you have this normally added cost of something like pesticide that you don't have to worry about. So those are the kinds of questions that I would want you guys to be asking. Now you guys ask some other questions about the slide presentation that these guys made. And they can be clarification questions. They can be any kind of questions you might have.
Okay, this is kind of just a generic kind of question. It could be dumb. So looking at everything kind of thus far and like what you presented on, there's not a major difference between GE modified crops and non-GE modified crops, correct? Is what this presentation is basically saying.
It seems
From
like,
the
at
sub-
least in terms of yield.
Yeah.
Is that where you were about to say, Mackenzie?
No.
No, that's not. What were you gonna say?
Well, so a lot of the genetically engineered crops that we're presenting in this presentation have very little or changes in them. When if you look at crops today, there's a lot more genetically engineering that goes into them. Like the information is good in this, but it's also kind of like older since it only goes up to like 2010 and we're in 2020. So just kind of put that in perspective, I guess.
So in what way are these crops less genetically engineered than crops today are? I'm confused by that.
Well, from what I understand, there's more like modifications in crops or seeds or whatever you want to call it today than what this study is showing. So like corn and maize only have the BT gene modified, and the soybeans only have the HR gene modified. When a lot of most seeds today have both BT and HR and maybe something else depending on what crop it is what I'm getting at, if that makes sense. The studies in this are only showing one modification in the seeds.
But I thought Blake said that maize and cotton here had both BT and herbicide resistance in them. Was that correct, Blake?
Yeah, in that experiment.
Okay. So
in my graph, that's what I was getting at. In my little section to read, my graphs were only BT.
Okay. All right. Okay.
That's what I'm getting at.
Olivia, did that answer your question sufficiently?
Yeah.
Okay. I was intrigued by, and I think Mackenzie, you were talking about these two graphs.
Yes.
Yeah. So graph one. So I get why BT adopters would experience a decrease in the amount of pesticides that they're applying on their fields, but why did BT non-adopters also experience a decline in pesticides that they applied to their fields? This is the type of question that you don't need a PhD to ask. It wasn't really explained when it was presented as to why both of these lines would eventually decrease, and it's a good question to ask. Why? So BT makes the crop insect resistant, but why would a person not using BT crops also apply less pesticide? Give it to us, Mackenzie.
Well, I didn't read that in the article, but I was like, depending on the seasons, if it's a very rainy season, it'd cause more insects, or even if the crops are close together and like say they accidentally breed, it could cause like a mixing in the crops as well.
So did they present any evidence that these BT traits were escaping out of the BT fields into non-BT crops?
They did not. So you could kind of assume that they did mix, maybe.
Yeah, so they didn't have any evidence to indicate that that would be the case, and it wouldn't affect those plants anyway. It would affect the next generation of plants if there was any interbreeding going on, and every farmer gets his seeds new every year. So they generally don't save seeds because these crops are hybrid crops. So everybody in the class, put on your critical thinking caps and ask yourself, why would both non-adopters and adopters decrease their amount of pesticide use? This is graph one that I'm thinking of. So imagine you have a bunch of farms. Each of these farms could be 1,000 acres, 1,500 acres, 2,000 acres. And you have one farmer who's using BT crops, which makes the plants produce their own pesticide. Another farmer is producing non-BT crops. Another farmer is producing BT crops. Why would, it's easy to understand why these guys would be putting less herbicide on their crops, but while they're putting less pesticide on their crops, but while they're putting less pesticide on their crops, these non-BT adopters are also putting less pesticide on their crops. Why would that be the case? And if I recall correctly, the report talked about this. Go ahead.
Are people from outside the group able to give a guess?
Yeah. I opened it up for everybody. Yeah. Although the people best equipped to give an answer should be the people who read the chapter. But yeah, go ahead.
I would assume it would have something to do with cross-contamination or like the imbalancing of the ecosystem.
What do you mean by imbalancing of the ecosystem?
So if there's a sudden change in one part of, because like this is assuming that all of these plots of land are part of kind of the same biosphere. If there's, and I don't know a lot about ecosystems. This is just going off my general knowledge. If there's a sudden change in one part of the ecosystem that is unfavorable to an organism, that organism will then seek out something that is favorable. And so if there's a reduction in pesticide in one sector, but not in the other, then the insects that are affected by that pesticide would then seek out the better area.
But if that's the case, then insects that are hanging out, insects that are hanging out over here wouldn't want to be here. They would want to be here, and so a non-BT farmer would actually be increasing pesticide use under that scenario, but they actually decrease pesticide use. So what does BT do to insects, Mackenzie, since this was your section?
The BT, it's essentially just kind of wards off the insects because it's like a built-in like type of insecticide, genetically engineered modification.
Do insecticides ward off insects, or do they do something else to insects?
Well, they kill them.
It kills the insects, doesn't it? So if you have a bunch of insects feeding in this field, and this field, what's happening in these other fields?
Less insect population.
Yeah, who was that?
Mackenzie.
Mackenzie? Yeah, exactly. If you have this field, and this field that are all killing insects, that basically means that the overall population sizes of insects are down. It's kind of like the agricultural equivalent of herd immunity. By knocking the infestation down in BT utilizing fields, you basically reduce the overall abundance of insects, and so you actually have fewer insects in the ecosystem overall. This is where I thought you were going to be going, Blair, and then you went off in a different direction. And so the overall population is lower, so everybody requires less insecticide because of those individuals who adopted the BT crops. And so there's this lag. So the interesting thing about this figure for me is that there's this lag. They're declining, but it takes the non-adopters a little while to realize that they're receiving the benefit of the BT adopters, and then you start to see that decline occur at a faster rate. And so you get out here to 2010, and essentially BT adopters and non-adopters are applying the same amount of pesticides to their fields. Does that make sense to everybody? Let's see, who did this one? Was this Branson?
Yeah, that was me.
Okay. Does anybody but me have a question about this figure? This figure confuses the hell out of me.
I'll be honest, I don't retain information well if there aren't words on the page.
Okay.
And so I'm going to have to go back and rewatch all of this to take notes on it.
So Branson, explain this to us again. What does this figure mean?
This is the usage of the herbicide per kilograms in the hectare.
Okay. So, and what is it showing?
Just the amounts that were used for those years in that size of crop, I guess.
So are those amounts increasing? Are they decreasing? Or are they staying roughly the same?
Well, I guess the soybean is roughly the same, but they all have a slight decrease until about 2001, 2002.
So,
and then they increase again.
But they're not doing this.
Right.
So they're fluctuating.
Yeah, they're staying. Yeah.
They're fluctuating, but there's not any kind of directional trend like you see.
Correct.
The directional
right.
trend in pesticides.
There's no big drop or rise.
Yeah, there's no big drop or there's no big increase. How does herbicide resistant as a genetically engineered trait, how does that work in plants in these crops?
It kills the weeds, but the crops are good without it. It doesn't kill the crops and the weeds.
So when you have a genetically engineered herbicide resistant crop, it allows you to spray herbicide on your crop and it will kill the weeds, presumably, and it won't kill your crop. So what would you expect to happen with herbicide application after the introduction of herbicide resistant crops?
I would expect it to grow pretty good if there's no weeds that are growing.
I'm not sure what you mean by that. This is the amount of herbicide applied to fields. This isn't anything about yield. What would you expect herbicide application to be after you introduce herbicide resistant crops?
You'd expect to use less herbicide.
What's that?
You'd be expected to use less of the herbicide.
Why would you use less of the herbicide?
Because if your plant's already genetically engineered to be herbicide
resistant.
Protected. Yeah, thank you. You'd hope maybe that with them being like that, I don't know. I just expect you to use less.
Plants aren't spraying herbicide. They're just resistant to it when it's applied. So
yes, they're resistant to it when it's applied. Yes, that's right, Blair.
You'd probably use more because I assume the weeds would also start to evolve to gain some herbicide resistance as a form of cross-contamination.
Yeah, presumably the reason you want herbicide resistance in your crop is so that you can use herbicide to get rid of weeds rather than tilling. Tilling, it loosens up the soil. You tend to lose topsoil. Tilling compacts the soil, which makes it more difficult for plants to grow. Tilling also opens up the surface of the soil so that you have to irrigate more. There are lots of negative consequences of tilling to get rid of plants. And so if you have herbicide resistant crop plants that you could just spray herbicide on and it doesn't affect your crop, but it kills all of the weeds, that means that you can do less tilling, which means it saves you some time because generally these herbicides are going to be laid down by a airplane in many cases. So what you should expect to see is actually not a decrease in herbicide use rather, but an increase in herbicide use. And what we don't really see, except maybe in cotton, is we don't see a huge uptick in herbicide use. So Branson, since this was your section, did the report say anything about why they didn't see an increase when they should have?
I didn't see anything, no. They just said that since there wasn't a fluctuation that they couldn't determine the risk for humans or the environment.
Yeah, and the people covering the next chapter will talk about that. But you're exactly right. They didn't present any explanation why they didn't see an increase when they actually should have expected to see an increase. And that was a little bothersome to me, why there wasn't more of an explanation for why there was no trend like there was for pesticide application when you should have expected a trend. Any other questions from you guys? So, Branson, you did the takeaways, right?
I think we all just kind of put our, Blake talked about them, but we all just kind of put our own little stuff on.
All right. Oh, so Blake talked about it, but all right. So for all of you, it says here since 1987 there has been a decrease in the diversity of crops grown in the U.S. Is that directly related to the presence of genetically engineered traits in crops, or is that just the continuation of what has been a long-term trend in decreases in crop diversity?
That has just been the continuation. So with every thing that they have, studied and came up with, that's what they've kind of decided the big average would be that there's really been no change.
Yeah. And then one other bullet point that I do have a question about is it says herbicide-resistant crops contribute better yield when weed control is greater than not. Somebody explain that bullet point to me. It's a little confusing the way it's written for me right now.
That was just basically the takeaway from the, I guess it was the graph. Let's see.
Hey, which point was that, Dr. Kolinsky?
It's this one down here. It's 1, 2, 3, 4, 5. It's the 5th point down. This one.
So that was their finding, that it contributes to a greater yield when weed control is better, or when it's improved because of the specific herbicides that can be used in conjunction.
Okay, so this is something that kind of recurs throughout this chapter that you guys didn't really key on very much. When you think about yields, they mentioned a number of times in this chapter that the use of both BT crops that basically create their own pesticide and herbicide-resistant crops, those yields oftentimes tend to be better when using those crops, but only in those situations where there's actually a huge pest problem for BT crops and where there's a huge weed problem for herbicide-resistant crops. And so even though there's this general lack of increasing or lack of a change in the rate of increase of yield, if you dig down into the data, what you see is that in areas that are being plagued by pests in a given year, and I'd have to go back and look in the report to figure out where exactly this was, in areas where they were having a lot of pest problems, BT crops actually did produce higher yields, but that got averaged out over all of these fields where they were gathering data from. And so the effectiveness of any genetic modification depends partly on what the conditions on the ground are. And so if you are in an area that's having a real problem with weeds, then herbicide resistance can help you because you can reduce those weeds and then they don't compete with your crop. In the same way, if you're in a really pest infested area, then having BT crops will increase your yield. But if you are in an area that doesn't have a lot of insect pests, having BT crops doesn't really affect your yield because the pesticides produced by the plants aren't really needed, in the same way that if you were spraying your fields, it's just kind of a waste of money because you're spraying your fields to get rid of pests that aren't actually there. And so there is a lot of kind of field specific effects that get kind of swamped out in these broader data sets. Does that make sense? And I think that's what, when you saw this in their main findings, that was what they were talking about, is this kind of field specific, farm specific, region specific sort of effect. Any other questions? All right, this last bullet point is more about the next chapter than this chapter, so we will just let the next chapter people handle that. All right. Next chapter is chapter 5. Jackson, Olivia, Alyssa, and Blair. You guys gonna share your screen?
I actually emailed you the PowerPoint and PDF.
Oh my gosh, are you guys just anti-screen sharing or what? I've shut my email down. What's that?
I think a lot of us took our notes on our computer so that we can face.
Oh, okay. All right, I get that. Then I'm gonna leave my email open so that I can retrieve these things as they come to me. All right. All right, can you guys see there? What are those pears? Are those pears?
Yep.
All right, take it away. Who's up first?
All right, I'm up. So hi guys. As a group, we were assigned to read chapter 5, obviously, and it is about the health effects in relation to GE crops. You can move to the next slide.
Yep, hold on, it's not responding to me. Okay, there you go.
Okay, so kind of an overview about what this chapter is about. This chapter looks at the different studies in relation to the safety of GE crops. They look at the testing efforts additionally, and it looks at the risk and the benefits associated with the GE crops. So they specifically kind of wanted to see if it affected human health, nutrition value, allergens, herbicide, and diseases. And so they were just looking at the effects in relation to any way due to GE modifications and engineering. So throughout this chapter, the committee kind of went through a process of testing and making sure that they were taking into consideration of either refuting or supporting hypotheses and claims in relation to GE crops, and if they were beneficial or non-beneficial. With this kind of like chapter and testing, you have to take in consideration there is limits into knowing what all the outcomes of effects of GE has on humans due to the timing of everything. Kind of looking at studies, you have to consider that you don't necessarily know all the outcomes yet due to evidence brought this far. Next slide.
It's not letting me advance to the next slide. Let me see what, oh, there we go. All right, is this the next one?
Yeah.
Okay, so the different testing that they do, the overview of the United States regulatory testing, the 3 categories are the acute test, and that's just like the effects on animals, and then the chemical compositional analysis, that's what the actual labeling on the products are and when they go to the store, and then the allergenicity testing or prediction, that's just the allergy and like the allergic reactions in humans and like what it would be. And it's based on coordinated framework for the regulation of biotechnology. The testing of crops containing Bt toxins they're already Bt testing of crops resistant to glyphosate and D2,4-D, sorry. New uses of herbicide, and the U.S. regulatory assessment conducted by U.S. and by EPA with new herbicide. Ready for the next slide too.
Alyssa, you're breaking up a little, so maybe move a little more slowly through the material.
Okay, sorry.
So I'm gonna go over the
Hear
first.
all that, or do I need to go over it again?
You're muted.
Like the 3 categories?
Did the rest of you catch that? I was following most of it, so I'm good. What about the rest of you? Okay, yep, we're good. We're fine, Alyssa. Alyssa,
Okay.
if you have another section, maybe turn off your video and that will help your bandwidth from where you're at. Go ahead, Blair.
All right, so I'm kind of going to go over the first 2 types of testing, and then Jack is going to go over the third. So the first type of testing is basically animal testing, and they started with rodents. That was like kind of with any scientific experiment, because rats are the closest associated with humans. They share a lot of similarities. That's why we test them. And there's really one efficient way to test, and that's when you add a large amount of a single chemical to their diet. In this case, it's Bt toxins. Where they run into problems are things called whole food tests. When they add these foods to their diet, they can't just add the chemical; they have to add the food. If it fits within the rat's diet, it works great. They replace the diet with about 10 to 60% of this genetically modified crop. If they don't, however, then it's really hard to achieve an accurate test because if it's not a part of their diet, because that crop in and of itself isn't nutritious to the rats. And so it's kind of counterproductive, and it'll show up with a lot of false data if they try and include that into their diet. So they did this thing called 90-day testing with Whole Foods. They did a bunch of studies on what is the most accurate time window to measure the effectiveness of these GE crops, and there was a lot of math done, and 90 days was the kind of standard that was set. You can go to the next slide. So there were a bunch of rat studies. It was really hard to condense all of these into a couple of slides, but they basically concluded that in the rat's diet, that there were not a lot of changes. There were a lot of changes. I'll kind of be going over the nitty gritty of it here in a bit. But after rodent studies, they moved on to the pig. The pig's used in many surgeries because some of their makeup is similar to humans. And the good thing about them is that most of their diet anyway includes these types of crops. And so it's a good baseline for which they can, brain isn't working. It's a good baseline for which they can measure the effectiveness without having to be too invasive into the pig's regular diet. Next slide. So this is where I kind of get into the nitty gritty. As all these tests came out, there was a lot of problems with these tests. This is where the chapter kind of qualifies itself and says like, hey, here's all the things that can go wrong with these tests. Food science isn't accurate. In fact, at the very beginning of the chapter, it says, we understand that food science is not a perfect thing. Just kind of take everything with a grain of salt. The problem with whole food testing in particular is that there's two big schools of thought in that. And that one, it wasn't sensitive enough to detect differences. They found that a lot of the stuff that a lot of the toxins that they were measuring didn't show up. So like if they put a known toxin, if they put a toxin in the food and they were like, we know this causes issues, let's see what it does, it wouldn't show up very much of a change, which would show that if there was a toxin that they didn't know if it affected them, it's inconclusive. Because of the amount of tests that they do in terms of Bt toxin on pesticides and so many other avenues, some would say it's unethical to do animal testing. The third part of that school of thought was the cost of 90-day tests. It is a big money suck for something that could not be beneficial. The second school of thought that they are beneficial, but they were concerned about the design, more or less like who was picking what was, I'll later talk about, biologically relevant. And in terms of this Paulson et al. Study, it was a study that was done in which the people, they did this study, and they said there's not a lot of difference in these statistics, in these GE crops and non-GE crops. What a lot of people found when they went back to this study is they found that there were actually a lot of significant differences, like the size of the rat's liver and kidney. But the scientists did not think that it was biologically relevant to include them in the study. And so a lot of people were going, wait, what's biologically relevant mean? There's no standard for that. And so that's where a lot of the inaccuracies in the tests came out. In terms of the multiple farm animal studies, they came to find that the GE crops and non-GE crops were actually bred in different locations, and so that creates a number of problems that aren't being seen. And the second thing was the amount of tests that they did. If you do more and more tests, it can come up with a lot of false positives, which doesn't display accurate information. Next slide. The second type of testing is compositional analysis. This one was less thorough because there's not as much that goes into it. It's basically what you see on the label when you turn around a box of rice. All these farmers submitted their nutrient chemical compositions of their plants. And again, there were statistical differences found, but they weren't considered biologically relevant, which again ran into a huge problem. The second big problem that they ran into was that most of these foods, when turned into the board for review, didn't show up a lot of negative effects. But then when they were put through heat, which a lot of people will do with these crops because of cooking and stuff, that it actually changes the makeup and the amount of protein and the amount of DNA in their section. And so it really messes with the study because of that inaccuracy. And so they started doing this new thing for substantial equivalence, finding out like how accurate they can get of these transcriptomics, proteomics, and metabolomics. Those are basically just enzymes that are in cells. Transcriptomes, proteins, and metabolomes.
Yeah, so I'm unmuted, right? So the transcriptome is looking at RNA that's floating around in cells. So the transcripts are just the parts of the DNA that actually get transcribed as you're going to make proteins. So that's a measure of like total gene activity. So you can look at the transcriptome. The proteome is then how much of that RNA actually gets translated into proteins. And then metabolomics is basically saying, well, what are the metabolic products that would come out of these metabolic processes? And you can actually measure those as well. And so they're looking at it from the standpoint of gene expression, proteins that are present, and then what the influence of those proteins are on kind of whole organism physiological function. So those are just fancy words for those things.
I tried for so long looking for the definitions of these. Couldn't.
Yeah, it's not easy stuff. I get it.
So the good thing about this is that it assessed a broad amount of characteristics, and it was more accurate than anything we've done before. And what they found is that even though there were some of these changes, the proteomes changed actually more than the other two. But even when those were detected, they themselves did not cause safety problems. Next slide. All right, and then Jackson will take it away for food allergenicity.
All right, so I have food allergenicity testing and prediction. The two ways of allergens that interact with the population is sensitization from initial exposure, elicitation of allergic response on the second exposure, and then mediated by immunoglobulins. The two scenarios of
Antibodies.
tests.
It's just a fancy pants word for antibodies.
Okay, so mediated by antibodies. Two scenarios of testing is where you transfer of any protein from a plant with known allergenicity, and then transfer of protein that could be a de novo allergen is the second scenario.
Okay.
Animal testing is not accurate. It is predictive allergenicity and it is insufficient. The likelihood of allergenicity, the higher in plants with known allergic responses, and higher likelihood of protein isn't dissolved by gut fluid. Next slide.
Did somebody have a question? I thought I heard somebody say something.
I was just asking if after the presentation you could describe a de novo allergen, because I again could not find that.
De novo, it means a novel, an unknown allergen. or a new allergen that's, so for example, a COVID would be considered like a de novo coronavirus because it's a coronavirus that we don't have any experience with. And so a de novo allergen would be a new allergen. So for example, BT would be a de novo allergen because it wasn't in our food prior to us putting it into our food.
And then food allergenicity continued. The gamazine concern, storage protein in maize, it's hard to digest, and the GE maize may have higher concentration, which is likely for allergenic. Young pigs develop antibodies against gamazine. There's no known difference in gamazine proteins. The connection between immune response and allergenicity, significant change in bacteria in the gut, no significant change in immune function. And then the treatment of acid reduction in children were more likely to have an allergy. Next slide. Some of the benefits of GE crops. There are a number of crops that have GE traits that can help improve human health, which would include commercialized and crops that are in the pipeline toward commercialization. Improvement of human health is the sole motivation for the development of a specific crop trait. And then crops with altered oil composition might improve human health, but all of that depends on the specific alterations, which would be like how the crops yield and how the products of the crops are used. The possibility of GE crops improving human health would depend on the exposure of plant-produced toxins to humans, and that would have to be lowered. But there's not enough evidence to see what the possibility would be. Next slide.
Okay, hold on, I'm taking a note on something.
So the concerns, they have been presented. Can you guys hear me okay?
Yep.
Okay, so they've been presented in regards to each other if not GE foods have like health effects. The type of cancer change over time, along with the patterns of cancer, the change is similar to the UK's, and the United Kingdom's diet contains lower amounts of food delivered from GE. So basically, they didn't find any evidence saying that like GE foods caused cancer. And the data doesn't correlate with the cancer rates. It's not supporting the claim of consumption of GE crops increases cancer rates. This far, no evidence that reports that GE foods cause higher obesity rates either, nor do they generate a protein that increases the opportunity of food allergies, bodily effects, or autism. And GE crops are not expected to cause health problems or substantial health risks. So basically, foods and the crops haven't caused any concern. I mean, they've caused concerns, but there's no evidence saying that the GE crops specifically caused the concerns. That was one. Next slide.
Okay, in conclusion, while testing was a big portion of our chapter, something to just consider is the process of testing of GE crops on animals may not be the most effective way of showing potential outcomes in humans with the future GE crops. Overall, it's supporting or refuting hypotheses and claims associated with the benefits of food derived from GE crops. While this is looking at the food and how the effects it plays on animals and humans, they're looking at the chemical composition of GE foods in relation to the consumption of it. There's no difference really in saying that there's a higher risk with eating GE foods versus non-GE foods. Something to look at as well is the time series of the data does not show that any disease or chronic conditions are correlated with the consumption of GE foods. The comparison of GE and non-GE plants are small and relative, and important to consider, but so far the evidence and studies aren't showing that there are major concerns presented. And another problem is to consider is when looking at this issue, you have to consider looking at the small versus the major changes in the genome. And that's kind of hard to look at when comparing it to animals and humans and the altercations with the GE cross.
And that's the end of your presentation, right?
Yes.
Okay, we're out of time, so we'll take questions on your presentation at the beginning of class on Monday. One of the things that I want you guys to be prepared for that I don't think you addressed particularly well in your presentation is you guys brought up the issue of doing animal testing is not necessarily the greatest way of perhaps testing for these health effects. But you didn't really go into a lot of the methodology of what the report did to try and work around these things. So they used a couple of techniques of looking at data data from humans, from populations of humans, that maybe allowed them to make some conclusions that something as simple as animal testing was unable to help them do. And I'm interested in you guys saying more about that, because I think it was an important part of the chapter that wasn't in your presentation, and I think your classmates would benefit from that. The last thing that I want to say, so be ready for that on Monday. The last thing I want to say is I have your exams graded. I'm going to be sending them out to you via email. Open the exam, open the email, because there are a couple of you that the exam was incomplete because of the program that you used to take the exam. And so I graded what I could grade of it, but I couldn't see parts of some of your answers because they were cut off by logos because you were using some shareware program, and it put logos and things like this over parts of your answers. And so I need you to look at that and then basically, retrieve that from your computer or come in and look at a hard copy version of it. Also, some of the questions were organized around tables, and it appears that some of you couldn't see the table on your iPad, and so it was a little gibberish and hard for me to make out what your answer actually meant. And I would rather grade your paper based on what you intended to write rather than me guessing what your intended writing was about. So expect those to be out in the next hour or so. And if you have questions about that, we can address those on Monday. But if it doesn't have a grade on it, it's because there was some aspect of it that I couldn't grade. And I'll try and put that in the email that I send to you individually. But in case I miss it, check for the grade. And if it's not graded completely, then you need to come and see me. All right. I'll see you guys on Monday.
Dr. Kowinski?
Yeah.
Could we have a quick chat?
Yeah, sure.
Okay. I have to run to the restroom real quick, but
sure. I'll hang on until then.